Hello everyone, and thank you for joining me. I'm Rabab. I'm here today to represent my presentation on metabolomics, methods to study metabolomics, importance and application of metabolomics. Let's move to the content of this topic, which are metabolomics, introduction, its history, methods which are involved to study small molecules, importance and application of metabolomics. Let's move to the next topic, which is on its introduction. The introduction is here to tell you that what we will learn from this topic. This assignment furnish a basic introduction of the rapidly emerging field of metabolomics. We learn about the principle of metabolomics study design, its purpose, application, some analytical techniques used in metabolomics. Let's move to the next slide. Now we will learn about metabolomics. Metabolomics is the systematic study of small molecules. It gives the whole profile of a molecule, including its phenotype, its structure, and metabolic fingerprint, which tell us about the chemical process in which a small molecule is involved. And it is target isotope based analysis of a group of small molecules. A small molecule can be a substrate or a product of metabolism which are influenced by both genetic and environmental factors. Small molecules are known as metabolites, and collectively these are known as metabolomes. Some examples of small molecules are sugars, lipids, glucose, amino acids as shown in this figure. Let's move to the next slide. If we talk about the history of metabolomics, then we come to know that in the beginning, Chinese doctors and ancient Egypt used body fluid to determine a biological condition, which is considered as the first earlier use of metabolomics. Sanctorius became the man who is considered to be the founding father of metabolomic studies. He used different instruments and he provided quantitative basis of pathology. Another man, J.J. Thomson, developed first mass spectrometer. Some other techniques were also developed by different scientists. Those techniques are nuclear magnetic resonance and chromatography. The first paper on metabolomics, though not called metabolomic at that time, was published by Robinson and Pauling in 1971. The name metabolomics was coined in the late 1990s. Now we will learn about the metabolome and metabolic reaction. Metabolome or small molecules interact with each other within and between biological systems and with the environment. Metabolic pathways are essentially series of chemical reactions catalyzed by enzyme whereby the product of one reaction becomes the substrate for the next reaction. These reactions can be divided into anabolic and catabolic. Now we will learn about the methods to study metabolomics. There are two main approaches which are used to design metabolomic study. These are untargeted and targeted approaches. The approach chosen will determine how you design your experiment, prepare your samples, and what analytical technique you use. Both targeted and untargeted key stages of metabolomic study follow a similar pipeline. An example of this pipeline for mass spectrometry based metabolomic study is shown in figure. In this, an overview of complete analysis of workflow is shown. It includes design experiment, sample preparation, direct infusion or chromatography, mass spectrometry, data acquisition, data processing, and last, analysis and interpretation. Now we will go through each of the main steps in more details. First thing in this workflow is design experiment. Design experiment in which the factors like sample size, randomization, and storage must be taken into account to guarantee reproducible and successful experiment that minimize erroneous variability and highlight the metabolite of interest and their potential interactions. It is essential to make sure that samples collected reflect and represent the biology in question. The second thing in this workflow is sample preparation, which involve some steps like collection, storage, extraction, custom preparation for individual measurement system. The third one key stage in metabolomics study is direct infusion or chromatography. The small molecule may be either directly or injected into the mass spectrometer through a coupled chromatographic system. Chromatography is an important step in metabolomics experiment to separate individual metabolites from a mixture. It is important to note that the most common technology used in mass spectrometry are gas and liquid chromatography. The fourth one key stage is mass spectrometry, which is a technique that is used in mass spectrometry based metabolomics. The fifth one key stage of metabolomics study is data acquisition, in which two most common techniques used, which are nuclear magnetic resonance and mass spectrometry. Mass spectrometry is an analytical technique used to measure small molecules. The analytes are ionized at ion source before they can be detected in a coupled mass detector. The resulting data typically consist of mass to charge ratio, time, and intensity triplets that describe for every detected ion mass, strength of the ion beam, and the time it is detected by the spectrometer. There are three basic things in mass spectrometry. First one is sample inlet, through which sample enter the mass spectrometer. Another is ion source, through which hard and soft ionization by creating high energy and low energy electrons, heavily fragments and few fragments compound are created that interact with analytes. And in last, mass analyzer generated ions are separated by their mass to charge ratio. For simplicity, charge is often assumed to be equal to 1, and this ratio is approximately equals the molecular mass of an ion. All mass analyzers exploit the mass and electrical charge properties of ions. They detect separate ions and they give the chromatographic profile of an ion, such as generated ions beam is recorded across multiple scans at discrete time intervals. If we talk about nuclear magnetic resonance, then we come to know that it is a non-invasive and non-destructive technique with a high reproducibility, making it a powerful tool for the searching of new and novel biomarkers by using magnetic field and radio frequency pulse. We investigate protons atoms from small molecules. We measure the resulting signals from small molecules within a magnetic field. We can simply say that this technique is used to detect metabolite and to measure organic and some inorganic compounds inside biological samples. Pro and cons of nuclear magnetic resonance include this. If we talk about the pro of nuclear magnetic resonance, then we come to know that this type of sample analyte is fast and all kinds of small molecules can be measured simultaneously. The cons of nuclear magnetic resonance are that this technique is less sensitive for metabolite detection. If we talk about the pros and cons of mass spectrometry, then we come to know that the pro of mass spectrometry is that it is more sensitive for metabolite detection. It takes less space and cheaper to buy. And cons of mass spectrometry are that the sample which are used in this technique must be derivatized before analysis, which increase the preparation time. Let's move to the next topic, which is data processing. The second last step of workflow of metabolomic study of a small molecule is data processing, which is used to extract biological relevant information from the acquired data. It includes many steps that are similar to mass spectrometry and nuclear magnetic resonance. A good understanding of the steps involved is important in order to minimize the risk of skewed or false results. Typically, the end studies is a feature matrix, a set of features as shown in figure. A feature matrix is typically a peak or signal that represent a chemical compound. A feature matrix contains the intensities or relative abundances of relevant signals for every sample, describing the metabolomic fingerprint. Ultimately, this feature list would become a list of identified metabolites with semi-quantified or quantified values. To compile a feature matrix, noise reduction and background correction are essential before feature can be extracted via peak picking. This process greatly ties up the data. Extracted features of individual samples are then aligned across samples to compensate for drift in the chemical shift or retention time mass spectrometry as shown in figure. Noise or error is an important consideration to factor in because it distorts the signal in your data. There are two types of noise in metabolomic studies. These are random noise and systematic noise. Aligned feature can then be aggregated in a feature matrix. A feature has a characteristic chemical shift. Chemical shift is a resonance frequency that can be used in column header. The rows represent individual samples. Let's move to the next topic, which is analysis and interpretation. This step is the last step of workflow of metabolomic study of small molecules. In this step, the most common statistical analysis approach are grouped into univariate and multivariate methods. In univariate methods, we take only one variable into account, resulting in differently weighted results. In multivariate method, we work on a matrix on a set of variables and highlights. Each method offers unique insight into the data structures. The goal of statistical analysis is the prediction of samples properties through generation of models that capable of the information contained in data matrix. In mass spectrometry, the mass to charge ratio and signal intensities are the two most important variables. In nuclear magnetic resonance, we select integrated signals of interest for data analysis. Without venturing too much into the area of statistical principal component analysis, PCA, as shown in figure A, are established method for multivariate analysis of metabolomics data. Principal component analysis is a method that enables us to reduce the dimensionality and to identify major trends and features, and it is also helpful to classify and to predict exercise. The quality of statistical models that are inferred depends significantly on the data processing, scaling, and normalization methods used. Therefore, successful data analysis requires careful investigation of multiple models for consensus building, such as don't rely on a single model. In the figure 10b, it is shown that a correlation heat map of a feature matrix typically used in metabolomics analysis Let's move to the next slide. Metabolomic resource at the EMBL EBI. Metabolomics related resources are available at EMBL EBI. The EMBL EBI European Bioinformatics Institute is an international governmental organization which is a part of European Molecular Biology Laboratory family focus on research and service in bioinformatics. EMBL EBI has three main resources that cover the field of metabolomics, as shown in figure. The first one resource is known as metabolites, that is served as a general purpose open access for metabolomics studies. Their raw experimental data and associated metadata. The second one resource is KeyBuy. It's a freely available database of chemical entities of biological interest. It contains manually annotated small molecular entities, molecules which are not encoded by the genome. The third main resource that covers the field of metabolomics is Reactome. It is a freely available curated database of pathways and reactions. We can browse it. We can gain a detailed view of the molecular process of human biology. Reactome do what? It represents biological process as a interconnected molecular event or reactions, primarily proteins and small molecules. Now I will tell you about the importance of metabolomics. Metabolomics plays its important role as an ideal tool for pharmaceutical, preventive, healthcare, agricultural industries, among others, biomarkers discovery, and Drug safety are two main examples where metabolomics has already enabled informed decision making. It plays its important role in improving treatment strategies. In upcoming days, metabolomics will make us able to trend of our own metabolome for personalized drugs. Metabolomics plays beneficial role as product and stress testing in food industries, control of pesticides to research in agriculture, in medical diagnosis, and in health care. Now we will learn about some of the applications of metabolomics. Certain significant role are played by metabolomics as in agriculture. It enables us to improve genetically modified plants, to develop new pesticides, to get the growing demands on farming. Another significant role of metabolomics is biomarker discovery. Biomarkers are small molecules that can be used to distinguish two groups of samples, typically a disease and control group. For example, a metabolite reliably present in a disease samples but not in healthy individuals would be classed as biomarker. Body fluids of organisms such as urine, saliva that contain highly informative metabolites can be analyzed through metabolomics fingerprinting. Metabolomic fingerprinting tells us about the biochemical process in which the metabolite is involved. And profiling phenotype of metabolites, or we can observe structure of metabolites for the purpose of biomarker discovery. Personalized medicine is another significant role of metabolomics that is used for quick medical diagnosis, to identify disease, to rapidly identify hundreds of metabolites, and to identify disease state. Other significant roles of metabolomics are it is involved in genetic disease testing, nutritional analysis, clinical blood analysis, clinical urine analysis, cholesterol testing, transplant monitoring, food and beverage testing, chemical shifting imaging, toxicology testing, clinical trial testing, fragmentation monitoring, drug phenotyping, water quality testing, and protochemical testing. Let's move to the next slide. This whole topic can be summarized as metabolomics is the large scale study of small molecules within cells, tissues, or organisms. There are two main approaches used in metabolomics study: untargeted and targeted. Methods to study metabolomics involve careful planning and study design, and two of the most commonly used analytical techniques are nuclear magnetic resonance and mass spectrometry. Metabolites, key and reactome are serve as three main resources in the field of metabolomics. this is the end of my presentation. I hope you enjoyed it. Thank you very much.